Welcome everybody um, to tonight's talk. This is Profitable Pastures, um, the um, the second run of it, and I'd like to thank our conference sponsors, Brussels Agri Service, Range Ward, and the Beef Farmers of Ontario. Also like to thank tonight's sponsor, the Ontario Sheep Farmers. Tonight, we're going to have the impact of grazing management on pathogen presence in livestock and the environment with Dr. Heather Murphy. Dr. Murphy has over 20 years in water research in both North America and abroad. Dr. Murphy obtained her PhD in environmental engineering from the University of Guelph. Following her PhD, she worked for the United Nations International Emergency Children's Fund, or as a lot of us know, UF, as a water, sanitation, and hygiene specialist in Mali and Madagascar. In 2015, she joined Temple University's College of Public Health, where she was an assistant professor for 5 years. In 2020, she joined the Department of Pathobiology at the Ontario Vet College as a Tier 2 Canada Research Chair and Associate Professor working in the area of One Health. She was recently inducted into the Royal Society of Canada in their College of New Scholars and was named one of Canada's top 100 powerful women in 2025. So with that, I will turn it over to Dr. Heather Murphy.
Well, thanks so much for having me today. I'm going to present on behalf of actually a master's student, Carly Dinga, and a postdoctoral fellow, Seijin Cheong, that did a lot of this work that I'm going to show you today. And I thought I'd just take a quick minute to explain a bit of my background because I come to agriculture from a very different background, and like I didn't grow up on a farm. I'm from Halifax, Nova Scotia. I did my degrees at Dalhousie University and then ended up at the University of Guelph where I focused on water treatment appropriate for developing countries. So that's a picture of me doing my PhD work in Cambodia in the middle of this slide. And then after that, I had no interest in academia and I really wanted to go and work abroad looking and improving water for those that don't have it. So I ended up at UNICEF and I spent 2 years working in Mali and Madagascar. And at that time, I got really interested in the public health implications of all the work we were doing. So, was the work that we were doing like impacting child health? And we were working with a lot of academics. And it kind of got me interested, maybe coming back to academia and interest in public health because I had been trained as an engineer. And so I ended up at the Public Health Agency of Canada doing a research fellowship where they asked me to quantify the burden of disease on Canadians in terms of drinking water. So, how many cases of illness does the average Canadian get from drinking tap water? And that was a newspaper article on the left that came out after some of that research was published. And then I ended up moving to the United States, to Temple University in Philadelphia, and ended up in a college of public health because the work I did was very kind of interdisciplinary, looking at water and human health. Um, and while I was in the US, I really wanted to come back to Canada, and OVC had this position open in the field of One Health. And at the time, I had no idea what that meant. And for those who aren't familiar, it means like the intersection of human, animal, and environmental health. And I read this job description and I thought, you know what? I kind of do that work. But I don't look as much at like the animal health side, but I look at organisms that can make animals and humans sick. And so luckily they took a chance on an engineer and hired an engineer in a vet school. And now I'm learning every day and learning about the agricultural sector. And I kind of dove in when I arrived at Guelph with the project I'm going to talk to you a bit about today. So I'm going to give you a bit of an outline of our study and like the methods that we use to collect our samples. Then I'll talk a bit about the results that we found in terms of pathogens that we found on pasture in soil and cattle feces, and the one that they can impact human and animal health. Then I'll talk a bit about our water quality monitoring results and then try to summarize kind of what farm management practices we found were affecting the pathogen present in water, soil, and animal fecal samples or cattle feces. So I'm pretty sure many people on this call are familiar with different grazing practices, but for the purpose of our project, we're defining conventional grazing as cattle that have unlimited access to land. They're not confined, or they might be alternately grazed. So they might move between two pastures. And then the farms that we studied that rotated, they had at least three or more pastures and rotated between kind of 1 and 4 days typically. So they moved in a structured movement through a series of paddocks. And so there is a fair amount of evidence that there's the benefits of rotational grazing include improved soil organic content, nitrogen fixing capabilities, improved plant distribution, and it avoids spot grazing where cattle just eat what they prefer on pasture. There's a little bit of evidence that it might be good for water quality in terms of improved stream bank stability and reduction of fecal coliforms and turbidity in water. There's actually quite little evidence in terms of cattle health. There's a bit on nutritional quality, and then we found one paper that suggested, well, maybe it would reduce pathogen exposure in cattle health. And so this research is driven by a couple of questions. The first is we want to see whether rotational grazing can improve soil health while improving animal, environmental, human health by one, reducing the presence and quantity of pathogens in livestock, and two, reducing pathogens and potentially antimicrobial resistant pathogens in receiving environments, which we defined as water, soil, and wildlife. And this study came about. I went on a farm tour shortly after arriving in Guelph in 2020 and spoke with several producers that were doing rotational grazing, and they wanted more evidence on the potential health impact of rotational grazing on their cattle health. And so these research questions are governed by a few kind of hypotheses. On a conventional farm, we hypothesized that really the feces is going to be more concentrated in certain areas based on cattle behavior. So in the farms that we visited, at least, we saw more concentrated feces in certain areas on pasture, on conventional farms compared to rotational, where it's kind of more evenly distributed because the cattle are being moved through these different paddocks. As a result, we think that we might see more die-off of pathogens in the rotational farms compared to the conventional, and less of those organisms will reach water. So more might reach groundwater and surface water in conventional farms compared to rotational. This is what we hypothesized. We also thought that perhaps the cattle might be interacting with each other's feces more often in conventional farms, which could be an animal health risk compared to rotational, where they're moving into a new pasture every couple of days. And by the time they come there, a lot of the pathogens might have died off or at least are reduced in the fecal material that remains. We also thought there could be impact on wildlife that resides in very close proximity to these farms. So this work was done in collaboration with a pretty large team. So we had Charlotte Winder, who's a ruminant vet, Claire Jardine, who's a wildlife vet, Carrie Dunfield, who's a soil microbiologist, Nicole Ricker, who's an antimicrobial resistance expert. We relied a lot on our producers, and then myself, I kind of wear this engineer, water microbiologist, public health hat. And what we were trying to do is really try to intersect the importance of animal, human, and environmental health through this project. So, what are the pathogens of interest? So, when I say pathogens, what do I mean? I mean, for this talk, bacteria and protozoa or parasites that can cause illness in humans and animals, so they can move between the two. So, the ones we studied here were pathogenic strains of E. coli, Salmonella, and Campylobacter. And then for protozoa or parasites, we looked at Cryptosporidium and Giardia. In terms of our methods, we compared the presence of pathogens across fecal, soil, water, and wildlife samples. And then we also had a detailed animal health survey that also incorporated feed changes and management practices that we gave to producers. We also collected rainfall data on site at all the farms. So our sample collection started in 2022 and went to 2024. We had some data from 2021, but that was more of a pilot study to try and figure out our methods and our approach. So the focus of this presentation is on 2022 through 2024. During that time, we sampled 10 farms. We collected 648 soil samples, 515 fecal samples, 84 surface water samples, 103 groundwater samples, and 65 passive samples, which were taken also from surface water. And I'll explain what those are in a minute. In terms of the field sampling, we had 5 rotational farms and 4 conventional farms. You'll notice that that number's dropped to 9 from 10. So we did have 10 farms, but we had one farm that had multiple different animals on farm, and in the end, it was a big outlier in analysis. So I'm only going to talk about the 9 farms that we ended up analyzing the data for. We collected biweekly to monthly samples, and we studied 3 grazing seasons from May to September. On each farm, we collected 5 unique fecal samples at every visit. And then for soil samples, we would do soil transects. And so what we did was we would do a transect where we walked along 3 different points and collected 15 samples along those points to represent a certain portion of the field. And then we took that sample and mixed it all together. So that would be one sample. And we collected samples where cattle were and where they were not on the fields when we arrived. So to try and represent their exposure to potentially feces or what their environment is. So on a conventional farm, we would take samples where the cattle were when we arrived and where there was evidence of a lot of fecal material. And then we would take samples where we did not see a lot of cattle, evidence of cattle, I guess, presence. And then for soil samples on the rotational farms, we collected from where the cattle were when we arrived, or if they had just left a paddock, that paddock, and then we would collect from the paddock that they're about to go into. In terms of water samples, we collected 1 liter grab samples from groundwater, so a well if that was on site, or surface water, and we analyzed them for what we call traditional indicator organisms, so E. coli, total coliforms, and fecal enterococci. And I'm going to explain in a bit what those are. Then we also concentrated large volume samples to be able to detect pathogens in water. So we would concentrate 50 liters of surface water or 800 liters of groundwater on an ultrafilter. So it's this kind of filter you can see on my screen. Um, it's like actually a dialysis filter. So we pump water through and we have a flow meter so we know exactly what volume has pumped through that filter. And then we take that back to the lab, we flush it and take everything that has been collected on the filter and then concentrate that down and then look for pathogens in that sample. We also deployed passive samples. So these are like little boats that we would put out in surface waters on farm or like so if they had a pond or a stream. And we have little filters inside. And what happens is those stay in the water in between our sampling times to hopefully capture contamination that we might have missed because we're only collecting like a snapshot in time. So these allow us to understand what might have happened between our 2 sampling points. In terms of how we detected pathogens, we use what we call a qPCR-based method, which basically looks at the DNA of the different organisms. And so we looked for 15 different genes that could represent those 5 pathogens that I talked about earlier. And so each one of these genes we ran 3 times to make sure that if we had a detection, we were certain that pathogen was detected. So these genes represented again Cryptosporidium and Giardia, which are parasites. Campylobacter pathogenic E. coli. We did Shiga toxin producing gene, which is associated with pathogenic strains of E. coli and Salmonella. And then we also tested for a fecal marker called ruminant bacteroides, which is basically a bacteria that's shed in high concentrations in cattle feces. So if we find it in water or in other places, we know that there's evidence of cattle fecal contamination. In terms of the microbial indicators, we also measured them in fecal samples. So we looked at fecal enterococci and did what we call serial dilution. So we took a sample and we diluted it, and then we grew up this bacteria on plates and then counted it. And fecal enterococci can be pathogenic, but more if we see a lot of it in cattle fecal samples, it could be an indication of could cause poor health, and they could be contaminating the environment because a lot of these organisms can be problematic for humans. We also looked at antimicrobial resistant E. coli in the fecal samples. And this is a problem because if we see antimicrobial resistant E. coli in fecal samples, it means that that resistance could actually be transferred to other organisms within a cattle gut. So if we're seeing that there's resistance in there, it can cause problems like for if you want to treat livestock for um, with antibiotics, they might not work if they've got resistance within their body. So we measured this by looking at antimicrobial resistance E. coli specifically. In terms of our farm management survey, we collected data on year and month, farm use, how many years the farm had been used, prior land use, farming practices in terms of numbers of days grazing, paddock resting time, if it was a rotational farm, stocking density, which we measured as the amount of cattle within either the full pasture, if it was a conventional, or within a given paddock for rotational. Cattle origin as well as cattle age. We collected data on diets such as corn, creep, hay, grain, and I think there was garlic and kelp and kind of natural minerals that were also that data was collected. And then in terms of animal health, we collected data on whether antibiotics were ever given to treat sick animals, vaccines, and deworming. We also deployed these rain gauges on all our farms so we could estimate precipitation 24, 72, and 168 hours prior to sampling. So what did we find? I'm going to go through what pathogens we found in the soil and cattle fecal samples. Um, And so our first goal was to characterize the pathogen present, and then also we wanted to look at that fecal enterococci concentration as well as the antimicrobial resistance E. coli in fecal samples. So I'm going to try and walk you through those. So in terms of the pathogens we found in fecal samples, Giardia and Cryptosporidium were the organisms we found in most samples. So you can see as well as Shiga producing one gene. So you can see we have kind of 77% to 98% of samples were positive for these organisms. So definitely shed in the livestock. They can be problematic for human health, but Cryptosporidium, particularly in young calves, can cause scours and health problems as well as some of the other organisms. So you can see we had also pathogenic E. coli was found in around 63% of samples. Now, in terms of like direct comparisons between conventional and rotational farms, you can see that in terms of like the shedding of organisms, we don't see huge differences. So this is an example of two farms that were paired by region. So this is a conventional and a rotational. On the x-axis is the sampling date, and then on the y-axis is pathogen presence. So 0 to 6 means like if we had one pathogen present, it would be 1. If there was two pathogens present in the samples, it would be 2, and so forth. So you can see, and each color represents a pathogen. So the dark green is Cryptosporidium, which is always present pretty much. And then we saw a lot of Shiga producing one gene, so associated with pathogenic E. coli. And then we also saw Giardia in a handful of samples. But you can see it's not that different between the farms. In terms of soil pathogen results, we analyzed them by where the cows were present at our sampling time and when they weren't present. And what you can see here is that we see a lot of Cryptosporidium in the soil. And so for those who aren't familiar, Cryptosporidium is actually a really hardy bug and it can overwinter. And so we think some of it, the presence of crypto was not necessarily related to recent presence of cattle, but it could be from prior years and an accumulation in the environment. Whereas the pathogenic E. coli would likely be a recent exposure because it doesn't survive very long in the environment, as well as the Giardia and some Salmonella. When we look at the cows not present, you can see Cryptosporidium is still there. And so that kind of a little bit holds true with we think that this old deposition of that organism keeps it survives and persists for long periods in the environment. So we're not seeing huge differences between when the cows were not present and the cows were present, but we do see a bit of a drop in the pathogenic E. coli between the two. And those ones were statistically significant for the pathogenic E. coli. Now, looking again at the pathogen presence in soil samples, comparing between the two types of farms, this is another example of a conventional 4 farm and rotational 4, and you can see Cryptosporidium is pretty ubiquitous across the two farms. In the conventional, we're seeing maybe a little bit more pathogens, but that's not necessarily statistically significant. And what did we see in terms of enterococci in fecal samples? So we looked at these are all paired farms. So on the x-axis, C1 is a conventional farm one, rotational is rotational farm one, and so on and so forth. And then on the y-axis is just the concentration of this bacteria we found in fecal samples. And we didn't find like a statistically significant difference between conventional and rotational farms. However, we did see overall slightly lower amounts of fecal enterococci in rotational farm cattle compared to rotational. The one or conventional, the one farm differences in region C, where the conventional farm has lower than the rotational farm. In terms of antimicrobial resistant E. coli, we saw similar trends to what we saw for the fecal enterococci. Again, the outlier was farms in region 3. So the conventional farm region 3 had slightly less amount of antimicrobial resistant E. coli in their fecal samples compared to rotational farm 3. Um, and we did see, as the grazing season went on, we saw spikes and higher concentrations, presence of antimicrobial resistant E. coli in fecal samples. So later in August and September, and we're not sure why that is. We hypothesize it might be because a lot of producers were feeding hay at the end of the season, and maybe it's something going on in the gut of the livestock. So moving on to water quality monitoring, we wanted to characterize the pathogen presence in water samples. And then we also wanted to characterize the presence of fecal indicator microorganisms in water samples. And we do that because then we can kind of compare it to benchmarks of what would be appropriate for like human drinking water quality and livestock drinking water quality, because we don't measure pathogens for that. We usually measure these indicator organisms, which I'm going to explain more in a minute. So I'm going to give you kind of a crash course in microbial indicators. So we have these groups of organisms that we use to measure bacterial contamination or microbial contamination of water sources. And you've maybe heard some of them before. So there's total coliforms, E. coli, and enterococci. And so total coliforms are bacteria found in excreta or feces of animals and humans, but they can also be less specific to fecal contamination. They can be from soil, sometimes can just contain total coliforms. But if you find it in water, it means environmental contamination. And especially in groundwater, if you see total coliforms, you know your groundwater is being contaminated by something. It could be feces, but it's something, or it could be something else in the environment. So total coliforms is the big group. Within that group, we then have E. coli, which is more specific. So E. coli is a type of total coliform, and it's actually under fecal coliform. So people have maybe heard of fecal coliforms, but that's measured less frequently now. So E. coli is a subset of total coliforms, and it indicates recent animal or human fecal contamination. So it gets more specific to say if that water has E. coli, it's likely got fecal material from humans or animals. Enterococci is in a similar group as E. coli. It can be indicator of animal or human fecal contamination, but it's more resistant to environmental stressors than E. coli, so it survives longer. So it might be more representative of organisms that can survive longer in water. And then within E. coli, we have the pathogenic E. coli, the one that's harmful to health. So just because you find an E. coli in a sample does not mean it's necessarily harmful to health, but it might indicate that you've got contamination issues and that a pathogen might be present. So to put this in context, the guidelines for Canadian drinking water quality put out by Health Canada is that you should have 0 E. coli and total coliforms in a 100 milliliter sample. So we should not detect it at all. There's no water quality criteria for beef cattle, but criteria for dairy cattle is coliforms should be really below one, kind of like humans, because coliforms greater than one can cause scours in calves, and coliforms greater than 15 to 20 colony forming units per 100 mils can cause diarrhea in adult cows, and cows may go off feed. And so right now there's no guidelines for beef cattle specifically in Ontario, but these are some numbers for frame of reference when I present our results. So what did we find? Pretty much every farm we had a well, and that was one of the water sources for cattle. And so in terms of the wells, you can see for total coliforms, E. coli, and enterococci, 100% of the wells were positive at some point in time for total coliforms, 45% for E. coli, and 100% for enterococci. Now we look at the actual samples because we collected 102. Of the samples collected, 52% were positive for total coliforms, 7% for E. coli, and 33% for enterococci. And then the concentrations, the mean concentration was around 32 for total coliforms, 0.36, or just less than one for E. coli, and 6.6 for enterococci. But these water quality represents water that's not really acceptable for human consumption and questionable for livestock consumption as well. And these were groundwater wells, and some of them were quite deep. And so here's just comparisons of what you can see between the different years over time. We didn't see a huge difference in the presence of these microbial organisms across the years in our study. We thought we might see differences based off of rainfall or other patterns, but we didn't. We saw some fluctuation in total coliforms, but nothing was statistically significant. And we have this line on here for kind of 15 colony forming units is what the dairy sector uses for total coliforms. And you can see that we exceed that in 2022, 2023, and 2024 on average. In terms of pathogens, what we actually found a lot of wells that were positive. And sometimes people are surprised when I show this work because I think people believe groundwater is free of microbial contamination, especially if it's a deep well. But Ontario has a lot of fractured rock, which means pathogens can rapidly transport into our aquifers and contaminate our wells. And so in our study, we found that 78%, or I guess 7 out of 9 wells were positive for cryptosporidium at some point, 4 out of 9 for Giardia, 6 out of 9 for pathogenic E. coli. We didn't detect any Campylobacter. We had 8 were positive for Salmonella, and then 1 was positive for Shiga producing 1 gene, and 9 showed evidence of cattle fecal contamination. In terms of samples that were positive, we had 9% were positive for cryptosporidium, 8% Giardia, 9% pathogenic E. coli, 14% Salmonella, and 2% Shiga producing 1 gene. And around 31% of our samples had evidence of ruminant Bacteroides. I wanted to give an example of what some of this water quality data might look like on a farm, because I think it's important for producers or anyone who owns a well to understand how intermittent microbial contamination can be. So on the x-axis is the sampling date, and then on the y again is the pathogen presence, which each block demonstrates a given pathogen. So you can see we had 2 sampling dates where we had pathogens that were positive. On those where I have these little cows show up, that's where we detected this ruminant fecal marker. So when we saw ruminant feces, we didn't always see pathogens, but then sometimes we did. Then if we pair this with the total coliform data and the E. coli data, you can see that on the sampling date where there was 4 pathogens present, we had positive total coliforms, but no positive E. coli. And then on the next date, we saw positive total coliforms, E. coli, and the fecal marker, the cattle fecal marker, and 2 pathogens. And then in 2024, we had ruminant Bacteroides, total coliforms, and E. coli, and no pathogens detected. So it's kind of to show that our indicators aren't that great and they don't correlate as well with pathogens as we would like. But in groundwater, in my experience, total coliforms has been a pretty good indicator of pathogens. Like if you have total coliforms in your well, you will see a pathogen at some point over time. And if you happen to take a sample and send it to an external lab and it was like in 2023 on on 0613, so the very first sampling point, and you came back with a negative result, you might think your well is actually free of contamination when in fact it could still be contaminated. And so all of the farm results looked very similar to this. Oh, and I forgot to add enterococci. So then you can see where the enterococci fecal organism shows up as well at different times. In terms of surface water, we had 6 farms that had surface water sources. 2 of them were sources of drinking water for cattle. 100% of them were positive for total coliforms, E. coli, and enterococci. And the samples almost like greater than 94% were positive for these organisms as well. And then the mean concentrations you can see were extremely elevated, so well above what would be recommended for dairy cattle, for example, as a drinking water source. We look at the indicator organisms over time. Or this isn't over time, sorry. These are the different types of water sources. So a rotational farm 1 had a large and small pond, and then rotational 4 had a pond. And then in the conventional farms, there was 2 streams, and 2 rotational farms had streams as well. And we do see a difference between, like, we had more indicator microorganisms in the conventional farms compared to the rotational, but conventional farm 3 allowed access to the stream. So we believe the cattle were contaminating that stream. And so it's maybe not a reflection of the grazing practices, but more just of that farmer who allowed access to the stream. In terms of surface water pathogens that we found, again, we found high rates of pathogens. So 100% of sites were positive for Cryptosporidium, Giardia, pathogenic E. coli, and Salmonella at some point. We had a couple sites that were positive for Campylobacter, and again, all were positive for this beef fecal marker. And then in terms of samples, we had 43% were positive for Cryptosporidium. That was kind of our max, down to Campylobacter was only 3%, and then the others were somewhere in between. But still, a lot of these samples were positive for organisms that can cause problems for human and animal health. And some of the streams were used downstream for recreational activities for humans. So that's a problem. Um, The next thing I wanted to talk briefly about was the presence of pathogens in ruminant Bacteroides. So we did a statistical analysis to see whether or not we thought that the cattle sources were associated with these pathogens. And so we ran kind of a statistical analysis to see when we detected this ruminant Bacteroides, did it agree with the different pathogens we found? And we saw some fair to moderate agreement that when we found the fecal marker that we found these pathogens, but we can't say for certain that these water quality results were indicative of the farm that the cattle were on, or if they were from upstream activities from other livestock, given these were really heavy regions where there's lots of livestock activity and manure being spread. Um, in terms of groundwater, the same thing applies, but we did also kind of see this kind of moderate to fair agreement that maybe these pathogens are associated with cattle fecal material. So what did we find kind of overall? And I had this more complicated, I guess, heat map showing all sorts of colors and what pathogens we found across all sample types. But I think it's a bit tricky to walk through. If anyone has any questions, I've got it at the end of my presentation and I'll pull it up. But I've tried to distill this down into big picture items that are more take-home messages that people actually use from our data. Um, what we found when we started analyzing things is that everything was very complicated and interrelated. And so what we wanted to do was try to identify common farm attributes that influence pathogen presence across our water, fecal, and soil samples all at the same time to be able to make some sort of recommendations. And in the end, we built some kind of complicated mathematical models that they actually were like little decision trees, and it kind of tells you in groups different attributes of on-farm activities and how that relates to what pathogens we found. And so, in terms of key findings, we found that pathogen shedding by cattle is complex and that the factors are all related. So we had these different kind of tree structures that came out of our work. And these were the main factors that influenced cattle shedding. So, what pathogens we found in cattle. So, the total number of animals. So, there's more animals, the more shedding we saw. Cattle age, total pasture size, stocking density, paddock size, total number of days grazing, supplemental feed. So, hay and natural feed like garlics and stuff seem to impact what we found in the cattle, deworming, and manure application. So if they applied manure on pasture. In terms of our findings in pathogens in the environment, we found like we couldn't directly compare rotational and conventional farms because I think we didn't have enough and there was a lot of variety within our rotational farm group. So they had a lot of different practices. But what we did see is that features of rotational grazing seem to be beneficial for pathogen reduction in water and soil. So kind of stocking density and things like that were coming out as being important, and those are features that are were more obvious in a rotational graze farm. So what we think is that there is reduced exposure to livestock, but what we couldn't prove is that this reduced exposure to their exposure in the environment equals improved health. So what were the most important factors? Well, we found paddock resting days. So the longer a paddock was rested, we saw fewer pathogens reaching water and soil, the number of days in a paddock, the number of grazing days, stocking density, as well as rainfall and soil moisture. We also found that our cattle drinking water sources far exceeded recommended guidelines for microbial water quality. And as I demonstrated, we found pathogens in all cattle drinking water sources, including wells. In terms of recommendations, we propose that reducing stocking density and increasing resting time may reduce pathogen presence and exposure. I know that this is a bit tricky in practice because those two things go hand in hand. So if you reduce your stocking density and increase resting time, it might mean you can have fewer like animals on pasture. But it is possible perhaps that you could graze multiple groups at different times to try and reduce the number that are together, but still maximize resting time. And for example, we had one farmer who really aggressively rotated and jammed cattle into very small paddocks. And we think that that wasn't maybe the best approach, and that really you don't need to jam as many cattle in, and you could have like a bigger paddock and maybe let them sit there for 2 days and then move on as opposed to going kind of daily. So we think there's a sweet spot. We can't tell you exactly what it is, but I would say 30 days seem to be a really good resting period from our data, and trying to reduce the number of animals in a given paddock. We also found that manure application might be problematic because we found higher concentrations of that fecal enterococci on farms where they applied manure. So once we talked to a few producers, some said that they didn't compost the manure very long. So they kind of have one composting heap and they keep adding manure. And then when it's time to put it on land, they put it on immediately. Whereas some of the pathogens probably didn't die off. So you're better off having multiple heaps where you kind of let it compost for 6 months to a year if possible before you then put it on pasture to try and reduce that pathogen load. We also recommend treatment and testing of cattle drinking water sources, as we found those high rates of pathogens. And one of our producers actually had a loss of a pile of calves, and the calves had scours, and they sent them off for fecal testing, and they were infected with the same pathogens we found in their well water. So, what were some of our study strengths and limitations? Strengths are that we did repeated sampling through 3 grazing seasons, and we studied a variety of grazing practices. We also did this on farm, studying real farms. So, as opposed to doing it in kind of a controlled environment, which is nice, but it doesn't always translate to the real world. We had farms matched by region to try and control for kind of soil and climate. In terms of limitation, we had small sample size for some of our variable categories, which made it harder for us to draw more conclusions. But I think we had to have this small sample size so we could collect lots of samples. So we knew what questions to ask if we were to do a larger study. So it allowed us to really characterize the systems. The other thing is we don't actually know if the pathogens we measured were viable, meaning they can infect cattle or humans, because we are only looking for their DNA. So it's possible that some of the organisms we measured and found is they were actually dead and we're just seeing their DNA, but we believe a large proportion of them would definitely be viable. In terms of next steps, we're in the process of analyzing wildlife sample data. We have been experimenting with some more modeling approaches with a colleague from the CDC who's got experience looking at farm management practices in the US. And we're trying to solicit feedback from producers and producer organizations to guide next steps or any additional analyses. We're writing up three kind of very academic papers, but we've got an infographic we're working on. But I'd be really open to any comments, suggestions from folks here. As I said, I'm new to the agricultural sector, so I'm here to learn from you as well. And we're trying to investigate the links between water quality and cattle health. So we're starting a study this summer. We're hoping to recruit 100 cow-calf farmers. So if you're interested in understanding relationships potentially between calf health and water quality, you can shoot me an email and I can tell you more about the study. And so with that, I want to acknowledge all the funders. So we receive funding from Beef Farmers of Ontario, the University of Guelph Food from Thought Initiative, through my Canada Research Chair position, the Canadian Foundation of Innovation bought my field vehicle. And there was an enormous team of students and staff from our lab on the right that went out all these years to collect all the samples. And I can't help but thank also all our producers that actively engaged in the project. They were fantastic. With that, I'll take some questions.
Thank you so much, Dr. Murphy. Yeah, so I guess my first question for you is what result most surprised you?
Hmm. Mm, maybe a bit that we didn't see a difference in the rotational and conventional, like straight out, because we did see evidence on land that like our hypotheses seemed true, and maybe it's just we didn't have enough sample size. So I was maybe a bit surprised on that, but I think we've pulled out the features of rotational grazing that still are important. But I also think it's a good story. One of our producers said, well, it's also, want to say that regular grazing is bad. Like that's not what we were trying to prove. We were also trying to just show like there are benefits of rotational grazing and it's not necessarily one or the other. And like if you can, however you manage your pasture, like even if you were to split up in four paddocks, maybe that would be beneficial and you don't need to necessarily aggressively graze. Yeah.
Okay. So our first question from the audience, this question asker has concerns regarding aerosolization of pathogens from manure spreading from neighboring farms. Are
Hmm.
any of the pathogens that you discuss likely to spread through the air?
Yes. I think the protozoa, I think it might be too big, but the bacteria, I'm pretty sure would be. And then there'd be viruses. We didn't look for viruses, but there's definitely viruses. They're a bit wimpier, so it depends on how long like manure sits for before it's being sprayed. But yeah, bioaerosols is definitely a concern, I would say, for like human health. There's a lot of people studying pathogens and bioaerosols from like downstream wastewater treatment plants and from agricultural operations.
Okay. Um, were any of the wells that you tested also used by the farm family as their drinking water, or were they only wells that were watering livestock?
All of them but one, I believe, was being used by the household. So we provided that information. And actually, a couple of our producers went out and bought treatment systems and started treating their water after we provided the results.
Okay. Um, this question comes from someone who transports water to livestock, and they've used chlorine tabs to treat the water.
Mhm.
If they follow the directions on the bottle of those tabs, is that enough to kill the pathogens that you've been talking about this evening?
Chlorine will work for the bacteria, but not the protozoa. So Cryptosporidium and Giardia are really resistant to chlorine. They're like a cyst, and the chlorine doesn't get in through them. So I typically recommend people use UV light as a water treatment option, and an inexpensive one, and it doesn't cause taste issues for livestock also. Chlorine is really effective, but it also depends on the source of that water. There might not be protozoa in that water depending on where it's coming from. Like if it's trucked water from already a treatment plant and you're putting chlorine to maintain it, that should be fine because it's already been treated. But if it's from a surface water or an unknown well water, it might not work if there's those protozoa in there or parasites.
Okay. Um, can you recommend a good sampling schedule for well water so that, excuse me, so that someone could get an accurate indication of the pathogens that might be in that well?
I get asked this question a lot. I assume every well is contaminated with a pathogen at some point. So the more we sample, the more we get a positive. And there's some research from the US that like in New Jersey, they sample like 20,000 wells and they have data that says once you're at like the seventh time, it's always positive. So I would assume that you're likely at risk. There's a handful of wells, I think, that don't never have contamination, but huge rainfall events, all sorts of things can introduce contamination in your well water. So sometimes I tell people for microbes, I'd say don't even bother testing. You should just treat. For chemical contamination, then you want to get it tested. And depending if it's like naturally occurring chemicals, usually it's pretty stable. So testing every few years is fine. It's not going to fluctuate unless there's like new industrial applications of things going on near your property.
Okay, thank you. Um, you mentioned that there are wildlife samples that have been collected and your team is working on the analysis. What kinds of samples were you collecting from wildlife?
So they did so Claire Jardine did small wildlife trapping. We trap like little tiny like mice and voles and things that live in close proximity. And her research has shown that like wildlife that lives in close proximity of farms is a good sentinel for like what's going on farms. So they trap them and then basically they get scared and then they poop. So we have the poop samples. And then some of them they collect like fecal swabs from the little creatures and then they release them. So we have those. They've been all analyzed for pathogens. We did get a pile of pathogens in them, but I haven't associated them. We haven't done any of the data analysis part yet. Mhm.
Okay, so something for all of us to look forward to is how might wildlife be interacting with this picture? Um, I smiled when you said it, but apparently you tickled our audience as well with scare the poop out of your test subject. But I'm sure they recover quickly once they're back in the wild doing their thing.
Yeah,
So, yeah. Um, I guess Heather, do you mind sticking your email in the chat for attendees just so that if anyone is interested in your upcoming project,
yes.
we can reach out to you for more information?
Yeah, or any questions really. I'm always happy to chat, especially about well water.
Because, yeah, since you're looking for cow-calf operations, um,
Mm.
some of our audience tonight are in that demographic. So if they're interested, please reach out to Dr. Murphy. Um, I guess I'll put out one final call for questions because I don't see any more in the chat right now, but just in case somebody is still typing, I'll give them another moment. Oh, there we go. So is there a specific test for protozoa?
There is not. That's the problem. All of these pathogens are really expensive to analyze for, and so that's why we use those indicator organisms. Um, yeah, so there's no easy test yet. And I think, and a lot of commercial labs don't do it either. So that's a tricky part. But if you have our cow farmer and you want water quality samples, that's what we're offering for the study. So you get water quality results back.
Very nice perk. So protozoa, hard to test for, hard to kill. But was your suggestion earlier of UV light? Does that work on protozoa as well, or is it just for the bacteria?
Oh, I see. It's Sandra. Hi, Sandra. It'll work on protozoa and bacteria. Yeah, and viruses. The only thing UV light is not as great at is one type of virus called adenovirus, which is a respiratory virus. It still inactivates it, but it's really hardy. But pretty much everything else dies off with UV light as long as you maintain your lamp and sleeve. So if the sleeve gets like covered in scale, it's not going to work. So you have to make sure that stays clean. But otherwise, it's a very effective treatment.
And by scale, you're talking like hard water, because I know
Yeah,
hard.
hard water or metals like iron and manganese in water can like form scale. So you have to keep it clean.
Okay, good to know because a lot of parts of Ontario have hard water, so
Yes.
maintaining water treatment stuff is an ongoing concern.
Mhm. Yeah, definitely.
Okay, well, I don't see any other questions in the chat, so thank you so much, Dr. Murphy. I'm going to turn things over to Joe to close us out.
Thank you very much, Dr. Murphy, for your presentation tonight. I think that gives a lot of people some additional things to think about. I'd like to thank our sponsors again for tonight, the Ontario Sheep Farmers, Brussels Agri Services, Range Ward, and the Beef Farmers of Ontario. Another thing to keep in mind, a heads up that the Ontario Forage Council is seeking to hire a summer student. If you're interested or know somebody that is, please contact the Forage Council for more information. And thank you.